Have you ever wondered what makes the human body function the way it does? The answer lies in the cells, tissues, and organs within our body that work together every second. A warm welcome to all of you from the Department of Nutrition, Sister Nivedita University. Today, in this video, we will explore all of these structures through the microscopic study of histological slides. So, let's begin by understanding what histology actually is. It is the branch of biology and medicine focused on the microscopic study of the structure, organization, and composition of biological tissues. Now let's all unlock the gateway to the fascinating microscopic world of histology. So what exactly is a microscope? It is an optical instrument consisting of a lens or a combination of lenses that produces magnified images of objects that are too small to be seen with unaided or naked eye. There are two types of light microscopes. One is simple light microscope and another one compound light microscope. A simple light microscope uses a single convex lens and visible light to magnify small objects and producing an enlarged virtual image. And a compound light microscope uses visible light and multiple lenses to magnify very small specimens such as cells, bacteria, and tissues that cannot be seen with the naked eye. A compound light microscope uses multiple lenses to achieve much higher magnification and better image resolution. This allows us to observe incredibly tiny structures such as cell organelles, bacteria, and other microscopic organisms that cannot be seen clearly with a simple magnifying lens. That's why a compound light microscope is preferred over a simple light microscope for studying histological slides. Before we move on to examining histological slides, let's first become familiar with compound light microscope. The compound microscope is divided into three major systems: the mechanical system, the magnifying system, and the illuminating system. The mechanical system supports the microscope and helps move and adjust the position of the specimen and lenses. It includes the base, arm, head or body tube, draw tube, stage clips, stage control knobs, revolving nose piece, coarse adjustment knob, and fine adjustment knob. The magnifying system consists of the ocular lens, also known as eyepiece, and the objective lenses, which enlarge the image of the specimen. Finally, the illuminating system includes the light source, condenser, brightness control, and switch. This system directs a controlled beam of light through the specimen so that it can be viewed clearly. With our microscope ready, let's together unlock the microscopic gateway to the fascinating world of human tissues and cells through the examination of prepared histological slides. So our journey begins with the histological slide of the kidney. It is being observed under 10x magnification. The presence of a cortex containing Malpighian corpuscles composed of a glomerulus and Bowman's capsule, proximal renal tubules with brush-border epithelial cells, and distal tubules lined by columnar epithelium are also visible, confirming that the specimen is of kidney. Now we shift our focus to respiratory system. First, let's see the lung. It is being observed under 10x magnification. As the identifying features of this slide include the presence of circular pulmonary alveoli lined by a single layer of pavement epithelial cells along with visible bronchioles and blood vessels. Therefore, the specimen is identified as lung. Now let's move on to the second slide, which is the trachea. We are also observing it under 10x. The identifying feature of this slide include the presence of hyaline cartilage, a mucous membrane lined by ciliated pseudostratified columnar epithelium, and the presence of muscle and elastic fibers. Therefore, this histological section is identified as the trachea. Now, let's shift our focus to the digestive organs. This slide is also being observed under 10x magnification. In this slide, we can see a mucosa lined with non-keratinized stratified squamous epithelium and a submucosa containing mucus secreting glands. The wall is composed of 4 layers: mucosa, submucosa, muscularis externa, and serosa. Based on these identifying features, the specimen is identified as the esophagus. The mucosa is lined by simple columnar epithelium. The lamina propria shows gastric peats and tubular glands containing parietal and chief cells. The wall is organized into 4 layers: mucosa, submucosa, muscularis externa, and serosa. Based on these identifying features, the specimen is identified as the stomach. Now let's move on to the small intestine. As we know, the small intestine is divided into 3 parts: the duodenum, jejunum, and ileum. Let's begin with the first part, the duodenum under 10x. This section shows the presence of long, broad, tongue-shaped villi lined by simple columnar epithelium. The submucosa contains Brunner's glands. The wall of the organ is organized into 4 layers: mucosa, submucosa, muscularis externa, and serosa. Next, we move on to the jejunum. We can observe the presence of cylindrical shaped villi lined by simple columnar epithelium along with goblet cells. The tissue also shows 4 distinct layers: mucosa, submucosa, muscularis externa, and serosa. Next, we are observing short leaf-shaped villi in the mucosa, which is lined by simple columnar epithelial cells along with goblet cells. We also observe the presence of Pierce patches. The tissue is also showing 4 distinct layers: mucosa, submucosa, muscularis externa, and serosa. Hence, the slide is identified as the ileum. Now in this slide, we can see the presence of folds of mucosa without villi. We also observe numerous goblet cells. The submucosa shows the presence of adipocytes. In addition, the muscularis externa is present, consisting of an inner circular and an outer longitudinal layer of muscle fibers. Hence, this section is identified as the large intestine. Now we can see the presence of a central vein. We also observe granular polyhedral hepatic cells radiating from the central vein. In addition, there is an intestinal connective tissue present. Hence, this section is identified as the liver. Next, we will learn the procedure for preparing a squamous epithelium slide before watch it under microscope. First, take a clean slide and remove grease by wiping it with 70% alcohol. Add 1 drop of distilled water on the middle of the slide. Now gently collect a sample by scraping the inner surface of the lip by the cover slip. Place a scraped sample in the water drop on the slide. Now add methylene blue stain to the sample and wait for a few seconds. If needed, remove the excess stain. Try to avoid the air bubbles. Now we will see it under microscope. Under 10x magnification, we can see numerous flat polyhedral squamous epithelial cells. The cytoplasm appears pale and slightly granular, and each cell shows a dark purple centrally placed nucleus. The cells are scattered singly and in small groups with minimal intercellular space. Finally, let's enter the last chapter of our journey, where we explored blood group determination and the microscopic study of stained blood smear with preparation. To determine the blood group of a sample, first clean the fingertip with 70% alcohol and allow it to dry. Using a sterile lancet, gently prick the fingertip to obtain a small drop of blood. Place a clean glass slide on the working surface. Carefully place 3 separate drops of blood on the slide. Now add 1 drop of anti-A serum to the first drop, anti-B serum to the second drop, and anti-D serum to the third drop. Using separate clean applicator sticks, Gently mix each drop to ensure proper interaction between the blood and antisera. If agglutination occurs in anti-A serum, it indicates the presence of A antigen, meaning the blood group is A. If agglutination occurs in anti-B serum, it indicates B antigen, meaning the blood group is B. If agglutination occurs in both anti-A and anti-B, the blood group is AB. If there is no agglutination in either anti-A or anti-B, the blood group is O. Now checking the Rh factor using anti-D serum. If agglutination occurs, the blood group is Rh positive. If no agglutination occurs, so it is Rh negative. Here, agglutination was observed with anti-B and anti-D, while no agglutination was observed with anti-A, which indicating a B positive blood group. Now, let's prepare blood smear. First, we are taking a clean grease-free glass slide. Next, the fingertip is being cleaned with 70% alcohol and allowed to dry. Using a sterile lancet, the fingertip is gently pricked to obtain a drop of blood. The drop of blood is then placed near one end of the slide. Now, second slide is being used as a spreader. Holding the spreader at an angle of 45 degrees, it is being drawn backward to touch the blood drop, allowing the blood to spread along its edge. Now the spreader is then pushed forward smoothly and steadily, producing a thin, even and tongue-shaped blood film. The smear is now being allowed to air dry completely. Next, 8 drops of Leishman stain is being added to cover the entire smear. The stain is left undisturbed for about 2 minutes to fix the blood cells. After the fixation, an equal amount of distilled water is being added to it and mixed gently. The slide is then left for 10 to 15 minutes to allow proper staining. Finally, the stain smear is being washed gently with the distilled water. Allow it to dry and is now ready for microscopic examination. Now, under 40x magnification, let's observe the blood cells clearly. These types of cells are called red blood cells. These types of cells are called neutrophils. These are monocytes and these are lymphocytes. Now, let's take a closer look at the morphological features of the blood cells we have observed. With all that, our journey through the microscopic world comes to an end for now. I hope this microscopic journey will remain a memorable experience for all of us. Until we meet again under the lens, keep exploring, keep learning, and stay curious.